After the process of transcription, particularly in eukaryotes, after the process of modifying the messenger RNA, we now go to the next stage of gene expression, which is the translation. If transcription is a DNA-directed synthesis, translation is an RNA-directed synthesis of a polypeptide. Again, translation happens when the genetic information flows from mRNA to protein through the process of translation. So, this an illustration again. Transcription happens inside the nucleus, and then once the mRNA in eukaryotes is processed, it will go out of the nucleus and go to the cytoplasm, specifically in the ribosome for protein synthesis. For the molecular components of translation, the tRNA, which is another type of RNA, or what we call transfer RNA, translates the mRNA message into protein. And these tRNAs transfer amino acids to the growing polypeptide in a ribosome. And then we will see that in a while. For this, you have here the ribosomal unit, and then these are the tRNAs or the transfer RNAs, which are associated with amino acids. And if you recall, amino acids are the building blocks of proteins. So when these amino acids form bonds between other amino acids, they will form a polypeptide chain, a long chain of amino acids. And this polypeptide chain is what we now call as proteins. So for the structure and function of the transfer RNA or the tRNA, always remember that each tRNA molecule carries a specific amino acid on one end and an anticodon on the other end. So we will discuss more of the anticodon and codons as we progress in this topic. So for the structure of the tRNA, this is a 2D structure of the tRNA. One end is carrying the amino acid and the other end is carrying an anticodon. And this anticodon is complementary to the mRNA strand that is carried to the ribosome. Aside from that, so here is just a 3D structure of the tRNA and a symbol used in our reference. So you have here the anticodon, the end that holds the anticodon or contains the anticodon, which is complementary to the mRNA strand, and the other end is an amino acid attachment site. So what do we mean by codons? So in translation, there is what we call or a code which is called the genetic code is widely used or universally used to determine the proteins that can be synthesized from a given mRNA strand. So for the genetic code, we have what we call codons, which is a triplet code, a series of non-overlapping three nucleotide words. So when we say nucleotide words, these are the nitrogen bases. And the words of a gene are transcribed into complementary non-overlapping three nucleotide words of mRNA. These words are then translated into a chain of amino acids forming a polypeptide. So here's an illustration on how the genes are translated into proteins. First, you have here a DNA template strand, and then it will be transcribed into an mRNA. So you have a single-stranded mRNA, and this mRNA, these are the words. One nitrogen base is equivalent to a nucleotide word. And these codons, they contain the three nucleotide words. They are called codons, and each codon will be specifying for an amino acid. So you have here three words or three nucleotide words which we call codons, and these specify for an amino acid through the process of translation. So a closer look on the flow of genetic information from DNA template strand, it will be transcribed into mRNA, and then this mRNA will be translated into proteins, which will enable the expression of the information dictated by our DNA. So one of the two DNA strands, the template strand, provides a template for ordering the sequence of complementary nucleotides in an RNA transcript. and the template strand is always the same strand for a given gene. So during translation, the mRNA-based triplets called codons are read in the 5' to 3' direction. And the other strand, if you recall, the DNA strand is double-stranded. Only one strand becomes the template strand, and then the other one is what we call non-template strand, which will be also called as the coding strand because the nucleotides of this strand are identical to the codons except that T is present in the DNA and play in place of U in the RNA. And each codon specified amino acid to be placed in the corresponding position along a polypeptide. So in the genetic code, there are 64 possible combination of codons, which was deciphered in 1960s. And out of the 64, 61 codons code for amino acids. Three of those triplets are stop signals to end translation. So just like transcription, there is the process of ending or terminating the transcription. Like in translation, it will be based on the genetic code, which will be triplets or the codons that will signal the end of translation. So the genetic code is redundant but not ambiguous. No codon specify more than one amino acid. So one codon is to one amino acid. And codons must be read in the correct reading frame in order for the specified polypeptide to be produced. Later we'll be discussing if there will be an incorrect reading frame, so there might be mutations or effect on the expression of the genes of the organism. So here is the sample of the genetic code. So you have here, as I have said earlier, each codon will specify one amino acid. So there will be no codons specifying two amino acids. There might be two codons specifying the same amino acid, but there will be no codons specifying two amino acids. And that is what we call the wobble position, which we will discuss later. So if in case you're given the mRNA strand, so this is our strand earlier in the previous video, how will you know the amino acids which will be produced by the mRNA strand? For example, in the first codon, which is AUG, you look at the genetic code. So in the left side of the genetic code, you have here the first mRNA base, which is A. And then at the top of the genetic code, we have the second mRNA base, you have U, and then the third are mRNA base, which is G. So you have here the amino acid AUG or the amino acid methionine. You don't have to write the start because usually in organisms, AUG signals the start of translation or it is the starting codon for translation. So you just put methionine on the amino acid. And then for the next codon, we have here UCU. You just follow the same procedure. Look at the left side of the genetic code. The first mRNA base is U. The second base is C. And then you have the third base, which is U. So this codon codes for serine or SER. And then you just follow the same procedure for all codons. And then you will end up at the amino acid sequence, which is methionine, serine, arginine, and alanine. That's how you do translation. It is based on the mRNA strand. And then, as I have said earlier, there are some codons which can code for the same amino acid. And the flexible pairing at the third base of a codon is called the wobble position. As you can see in the genetic code, there are some codons here that can code for the same amino acid. And the wobble position is that only the third base will be different. That is what we call the wobble position. So this will allow the amino acids or this will allow the codons to code for the same amino acid. But then again, there are no codons coding for two amino acids. The ratio will always be one codon, one amino acid. But because of the wobble position, there are there can be several codons that can code for the same amino acid. And since the ribosomes are the sites for protein synthesis, we have to know what is in the ribosome that makes it the site for protein synthesis. So ribosomes facilitate specific coupling of tRNA anticodons with the mRNA codons in protein synthesis. There are two ribosomal subunits, the large and small subunit, which are made of proteins, and the ribosomal RNA. So we already have discussed mRNA, we have here the tRNA, and lastly, the one responsible for protein synthesis is the ribosomal RNA, which is located in the ribosome. So bacterial and eukaryotic ribosomes are somewhat similar but have significant differences, which will be discussed in higher bio subjects. And some antibiotic drugs are specifically targeting bacterial ribosomes without harming eukaryotic ribosomes. So the ribosomes, there are the imaginary part, quote unquote imaginary parts of the ribosome. We have the P site, which holds the tRNA that carries the growing polypeptide chain. So if this is the ribosome, we have here the P site, the peptidyl tRNA binding site, which holds the tRNA molecule. The next site in the ribosome would be termed as the A site, which holds the tRNA that carries the next amino acid to be added to the chain. So you have here the P site, and then the next tRNA which will supply the next amino acid will be in the A site, which is the amino acyl tRNA binding site. The next site is called the E site, wherein the tRNA leaves the ribosome. So this is the E site. In other words, you can have here the acceptor site. You can use the acceptor site as a mnemonic, or just to remember that the A site accepts the next tRNA. While the P site, this is the holding site, and then the E site or the exit site, it will be the one releasing the tRNA from the ribosomes. And then this is just to show you how the translation progresses in the ribosome. As I have said earlier, you have here the P site, which holds the tRNA and the growing amino acid chain. And then the next amino acid is accepted or located in the A site, and it will go to the P site once this tRNA is done. And then once the tRNA have attached the amino acid, it will leave through the exit site or the E site. Okay, and then in building the polypeptide or in translation, we also have three stages: the initiation, elongation, and termination. And all three stages require protein factors that aid in the translation process. And aside from that, energy is also required for some steps. So in the initiation of translation, the start codon, which is AUG, as I have said earlier, signals the start of translation. First, a small ribosomal subunits bind with mRNA and a special initiator tRNA, and then the small subunit moves along the mRNA until it reaches the start codon. Proteins called initiation factors bring in the large subunit that complete the translation initiation complex. We have there the mRNA, and then the small subunit and large subunit, as well as the initiation factors that will form the translation initiation complex. So here is an example of the small ribosomal unit binding to the mRNA until it reaches the start codon. And then once the large ribosomal unit binds, so there is the translation initiation complex. In the elongation of the polypeptide chain, each addition involves proteins called elongation factor. So there are three steps in elongation: the codon recognition, peptide bond formation, and translocation. Energy expenditure occurs in the first end, third step. So during translation, it proceeds again in the 5' to 3' direction, and the ribosome and mRNA move relative to each other, codon by codon. So what happens during translation? You have here the first step, the codon recognition. So wherein the tRNA will attach to the P site and then deliver the first amino acid. And then once the tRNA attaches the amino acid, another another tRNA will come in the A site carrying the next amino acid, which is complementary to the existing mRNA bases. After that, the second amino acid will be joined together, or the amino acids will be joined together through the peptide bond formation. If you recall from your Gen Bio 1 class, we have the peptide bonds between amino acids or proteins. After the peptide bond formation, we have here the translocation, wherein the the tRNA carrying the first amino acid leaves through the E site, and then the next tRNA carrying the next amino acid moves to the P site, and the A site is ready for another tRNA. So that is the translocation process. Again, we have the codon recognition, the peptide bond formation, and translocation. For the termination of the translation, a stop codon is reached in the mRNA in the A site of the ribosome. So the A site accepts a protein called a release factor, and this release factor causes the addition of a water molecule instead of an amino acid. So this reaction releases the polypeptide, and the translation assembly comes apart. So in this case, you have here the stop codon. You can see in the genetic code, we have three stop codons, the UAG, UAA, and UGA, which which is a stop codon for the mRNA. So once the ribosome reaches the stop codon, it will have it will accept a release factor which promotes hydrolysis. So from your Gen Bio 1 class, hydrolysis would be the breaking of bonds in a molecule. So that hydrolysis process will cause the ribosomal subunits and the other components to dissociate. And the polypeptide is also finished by that time. So just an overview of the gene expression or the central dogma of molecular biology. We have here transcription in the nucleus, wherein it produces a premature RNA. And then this premature RNA needs to undergo processing through the 5' end capping and the poly A tailing in the 3' end. And mature R- mRNA and this mature mRNA will go out of the nucleus. It will go to the cytoplasm, particularly to the ribosomes. And the ribosomes, it has three sites, the E site, P site, and the A site. So the A site will be the amino acyl site, or the quote unquote acceptor site, which should accept the tRNAs carrying the amino acids. And then the P site will hold the the tRNA carrying another amino acid, and there will be a peptide bond formation. Once the tRNA delivers the amino acid, it will leave through the E site. Okay, so the translation, we have three processes: codon recognition, and then peptide bond formation, and translocation. So translation stops when the stop codon is reached. We have three stop codons: UAA, UGA, and UAG. What if an mRNA strand is read incorrectly, or the reading frame is read incorrectly? So mutations happen. When we say mutations, these are changes in the genetic information of a cell. And then when we say point mutation, this is a specific type of mutation wherein only one nucleotide pair of a gene is changed. The change of a single nucleotide in a DNA template strand can lead to the production of an abnormal protein. So if a mutation has an adverse effect on the phenotype of the organism, this condition is referred to as a genetic disorder or hereditary disease. There are different types of small-scale mutation. So the point mutations, we have the single nucleotide pair substitutions and nucleotide pair insertions or deletions. So during substitution, there is something called nucleotide pair substitution, which replaces one nucleotide and its partner with another pair of nucleotides. And then silent mutation, they have no effects on the amino acid produced by a codon because of the redundancy of the code or the wobble position. And then during missense mutation, uh, the nucleotide pair substitution will still code for an amino acid but not the correct amino acid. And in nonsense mutation, instead of having another amino acid, it will code into a stop codon, which will lead to a non-functional protein. And then during insertions and deletions, they are just additions or losses of nucleotide pairs in a gene. So these mutations have a disastrous effect on the resulting protein more often than substitutions do. And aside from that, it produces a frame shift mutation. So frame shift mutation happens when the reading frame are altered or when the reading frame is altered. And that would be all for our lesson 2 of module 2 for central dogma of molecular biology. Thank you for listening.